So I think what we have yet to explore are what are those subtle factors out in the environment that may affect why genes become one protein versus another, and that's way down the line.
Replacement gene therapy involving the introduction of new genes will continue to carry risks until we know exactly how genes work. But new research here at the University of York could prove much safer. Instead of replacing a faulty gene, it destroys its message by attacking the messenger. It's the first step in tackling the biggest killer of all, cancer. This is the nucleus of a cervical cancer cell that has been forced to self-destruct. The cancer is caused by a sexually transmitted virus called HPV. Every year, 400,000 women across the world are killed by it. Medical pioneer Professor Joe Milner has spent 3 years trying to find ways of defeating it. Inside the nucleus of a healthy cell are the genes P53 and RB. These two genes suppress tumor formation by controlling the rate that our cells divide and destroying any damaged cells that might become cancerous. Cervical cancer occurs when the cancer virus enters the cell and attacks the tumor suppressor genes by placing two of its own genes, E6 and E7, into the human genome. These take over the reins of cell growth and force the cells to divide in an uncontrollable manner, causing cancer. Professor Milner's study, at the cutting edge of genetic research, is called RNA interference. It treats the cancer at its genetic root.
Conventional treatments of cancer, including chemotherapy and radiotherapy, are effective for various tumor types, but have the disadvantage in that they also damage normal cells. In other words, they're non-selective for the cancer. The huge advantage of RNA interference is that it can be tailored to selectively target cancer cells.
Genes are read by messenger RNA, which takes their instructions to the protein-making factories in the cell. Professor Milner's technique prevents the message from the cancer genes reaching these factories. It stops the message by effectively shooting the messenger. The agent that does this, a molecule called short-interfering RNA, is introduced into the cell that's been infected by the cancer virus. Here it combines with the messenger RNA, carrying the virus's cancer-making instructions to the protein factory, and destroys it, causing the cancer cells to self-destruct. On paper, it's an amazing concept. The question was, could Professor Milner get it to work in practice on these killers, the cervical cancer cells?
You can see that the picture immediately is different. There are far fewer cells, and here the cells with halos around them. These are cells which are in the process of committing suicide, essentially. These are dead. These are the remnants of cells, of the cancer cells. It was a wonderful feeling looking down the microscope and seeing these results. You could see that those cells were dying, whereas normal cells, which had had exactly the same treatment, were completely unaffected.
Now we've got a technology which allows us to interfere directly with the actual RNA genes of the virus. And certainly in its early days, the progress has been quite remarkable. So what we might have is a new generation of medicines.
From Professor Milner's research, you might think a cure for cervical cancer was imminent, but human trials are at least 3 years away. Our poll has found overwhelming optimism that genetic science will bring cures for most diseases. The British and Americans are more skeptical, but even here, more than 3 out of 5 people believe genetics will cure disease.
I think genetic manipulation, not at conception, but past that, is going to be the biggest course in medicine probably in the next 20 years.
Hopefully the developments in genetics will continue to improve our lives.
Gene therapy and research like Professor Milner's offers hope to an end to many genetic disorders where only 1 or 2 genes are involved. But in other big genetic killers like heart disease, A complex combination of genes is at work, and a different approach is needed. Radical techniques using the very latest genetic research hold great promise, but they bring a whole new set of ethical questions.